At 5:02. All right, there is a formality that awaits. I've announced Francis Chung's presidency twice already. Francis tells me I have to announce it a third time, so I will. I can't say it often enough. So Francis, congratulations, and we're a very lucky society to have you in charge.
So David, I would like to present David with a small token of gift for his excellent work and also for his leadership and also for his level-headedness. So he has provided really and contributed to SESM as the founding president for the last 3 years, and we are truly grateful for his leadership. Thank you again. So I would like just about 2 or 3 minutes to talk about our incoming terms as my president. First, I would like to thank all of you that you are supporting SESM. Anesthesia is sleep, and sleep is anesthesia. It's closely interrelated, and that's the vision of the Society of Anesthesia and Sleep Medicine, to bring the sleep medicine specialists, to bring the basic scientists people, and also for anesthesia together in one room to interact and also to flourish. And we have established that in our last 3 years, our annual meeting. And this comes as a sleep apnea special interest group. So what we are going to do now that we have a solid foundation of annual meeting, CME effort in terms of our newsletter, in terms of our literature update, and also feature article. And we also have to thank Sammy to provide us with very solid organization support. And I have to thank Marie. Marie, please stand up. So Marie actually has provided, again, the administrative support for SESM, and without her, we cannot be anyone or anybody. So thanks, Marie, to you. Now, what we'd like to do in the next 2 years, I would like to say that we will soar like an eagle. And what we are going to do is to establish 3 areas. One is the basic science, which we will establish special interest group to attract all the basic scientists. The other group is the pediatric sleep apnea group, which again is a very important factor in also pediatric sleep apnea and anesthesia, a major problem because FDA actually identified in February 2013 that codeine should not be used in children with tonsillectomy. And the third group that is very important is the ENT OSA, because surgery for sleep apnea is a key issue. And in 2001 trial, there was a big medical legal article that actually told us about 56 cases of sleep apnea for surgery, and they all have mortality and great morbidity. So again, it is a group that we need to interact with and bring them in. So the ENT surgery, they want to do it as outpatient, but they are deaf. So how can we incorporate the science and make it a fact? And my vision is that malignant hypothermia in the 70s is a big issue. Everybody is afraid of malignant hypothermia. But with knowledge, with research, and with education, it is no longer a catastrophe. Why? Because we know the genetic, we know the history, and we know the treatment. Sleep apnea will be the same. With malignant hypothermia, we actually say, we ask whether the patient has an issue with anesthesia, past history. We also asked a family history of anesthesia issue. Those two questions help us. So the undiagnosed sleep apnea is a major issue. Can we do the same? Dantrolene, genetic, are the things that will help malignant hypothermia firmly on the ground so that we don't have that. At the present, most of the things that we recommend on sleep apnea anesthesia are actually opinion-based with no scientific support. So how can we get there? And we have to rely on you. So thank you very much for your support.
We had an excellent positive feedback about the sessions yesterday, and of course we're into the entree today and looking to another very productive and exciting day ahead, and we've got a very interesting program organised for you, and so I'm not going to spend a lot of time preambling. We'll get into that in a moment, but I do want to make the point that meetings like this would not exist without the generous support of our sponsors, and I do want to, it's not an endless list, so I do want to mention them specifically. At the top line are our platinum sponsors, Philips, Respironics and ResMed, without whose support we'd struggle. So I want to particularly thank them. But also Massimo, very important people to us, Asa Covidin, Neon Coden, Fisher & Paykel Healthcare, Respiratory Motion, Hospira, Curex Pharma and Sleep Image. And they're all exhibiting downstairs, so I do think that we should it's important to us and important to them that there's an engagement there, and so I hope that at our breaks and at lunchtime that you do engage with what they've got to show you, because they've got some very important products, and they're on the translational end of all our research efforts, these companies, and so they're there to be encouraged. I'd like to hand you over to Babak, who's got a couple of announcements to make, and then we'll get into our meeting.
Thank you, David, and welcome everybody. As you all know, this is our third annual meeting of the Society of Anesthesia and Sleep Medicine. And before we start with our keynote speakers, I just want to make a couple of announcements. As you will see in your book that has all the abstracts and everything in it, just before the abstract table of contents, there is a little paragraph about CME credits, how you can get your CME credits. I just want to call that to your attention because it's online, and you can start at the end of today, and you only have until November 11th. So if you want to do it after November 11th, you won't be able to get your CME credits. So I encourage you to do it if you're interested in it. And also, you will find in your package a sheet like this. So if you get signatures from all the sponsors downstairs, hopefully you'll listen to them and not just go and ask for a signature. Then you can be entered in a raffle for a mini iPad. But don't work very hard because it's already been selected. I think I'm going to take it. You know, I'm from Chicago. It's been decided already. But all kidding aside, I cannot enter in the raffle, which is upsetting. But so without further ado, I want to introduce our first speaker. This year is exciting because we have two keynote speakers. Dr. Rich Horner from Canada and Dr. Javaheri from Cincinnati. So this is great, and both of them are leaders in the field. Dr. Horner is, as you can see, Professor of Medicine and Physiology at the University of Toronto. He's had an illustrious career. His research in sleep and upper airway neurobiology has been funded by the Canadian equivalent of our NIH for many years now. And you know, he has leading roles at the university. He's director of sleep and biological rhythms in Toronto. So we're very excited to have him here join us and give us his talk. Thank you.
Firstly, it's my pleasure and my privilege to be here today. I thank Babak, as well as David and Francis for the invitation. It's a double privilege because I've been able to catch up with some old friends I haven't seen for a long time, and also importantly to meet new people, and especially to meet people whose names I had really only associated with a paper that I've been stimulated by. So it's a great honor to get to meet people and get together. Firstly, I'd like to start and end with do we have a pointer, by the way? With my collaborators. The first three listed here from the University of Toronto, and some of their work I will be presenting. Also like to acknowledge Beverly Orser, who's becoming an increasingly important collaborator and partner at the University of Toronto, and some other collaborators that I will list here and whose will come up at different places with respect to the work I'm going to present. Thinking about disclosures, in June of 2011, I was at a 2-day session at Galleon, and between 2010 and 2012, we had an academic collaboration with Lilly Research Laboratories in the UK. This to me is an equivalent to having an academic relationship with another university. There was no personal financial gain, no honoraria, no protected findings. It was more a meeting of the minds with different expertise, and our collaborating partner was Stuart Hughes, who was an expert in channel biology, which we were not. And essentially the outcome of that collaboration was basic science knowledge. We published 4 papers together, so it's really to spur discovery science, but I mention it for the sake of openness. And the funding of our lab has been through the Canadian Institutes of Health Research, as Babak mentioned, but also the Canadian Lung Association through the Ontario chapter. The talk is outlined into 2 main sections. I'm going to initially focus on opioid-induced respiratory depression, mentioning what we consider to be one of the key sites for how opioids depress rate and cause central apnea. I think an important issue is the state dependency of that response. But I think this is very current data, which is in submission, is how are these opioids actually acting at the cellular level to depress breathing? I'd then like to move from respiratory rate to the effects on the upper airway and the sites for opioid-induced upper airway motor suppression. Then I'd like to switch to how sleep mechanisms cause profound motor inhibition to the upper airway, and then bring in a new concept with respect to tonic inhibition and the role of extrasynaptic GABA A receptors. I put this talk together afresh for this meeting, so I'm unsure about time, but I'll keep an eye on it for the purposes of keeping into my allotted time. If I have time, I would like to introduce 2 slides at the end about a new established program in sleep, sedation, and anesthetic sciences. And I think this, to me, is one of the exciting things that's developed over the last few years. We weren't ready for this, I would say, 5 or 10 years ago. We were aware of the importance of the subject through Ralph Leidick's leadership. He's one of the pioneers in bridging fields of sleep, sedation, and anesthesia. And as time has gone on, we've positioned ourselves now to really take advantage of these links. And I think the fact that it's happening at the society too, we're like a microcosm of this society and our own institution, and it's a very exciting time for us. And many people you'll recognize in the last slide, Francis included, who are involved in this program, which is established only in June. This is very much a data-driven talk, so I'm not going to provide an exhaustive review. There are other reviews. I know this is a small projector, but I can make these slides available. But I really want to emphasize some new findings in science that I hope you can apply to your either basic science or clinical backgrounds. So I'm just going to introduce the respiratory network. The respiratory network, the details of the respiratory network are complex. From the control of the primary or the diaphragm muscles of breathing, and also the secondary or the accessory muscles of breathing, including those that drive the upper airway. Also complex is their wiring in the medulla, This is a detailed diagram, but really what we will tend to focus on as time goes on and be explained in more detail is this particular circuit. The cells in what's called the pre-Botzinger complex that drive through interactions with cells in the reticular formation, that drive the muscles of the tongue, which influence the upper airway, and the cells which are thought to be the source of rhythm generation and impart rhythmicity to other components of the respiratory network, which ultimately drive the respiratory pump muscles, in particular the diaphragm. And we're going to talk about how it is that these cells are influenced by sleep and how they're influenced by drugs which can cause respiratory depression. So what we want to, this complexity ultimately produces the stereotype patterns of activity that are present across sleep-wake states in the upper airway muscles, the diaphragm muscles, and other muscles of the chest wall. We can distill that complexity, I think, into three essential elements. The controlling elements that drive the musculature of the upper airway, of which the particular focus today will be the hypoglossal motor pool. The control of the motor output, the rhythmic motor output to the muscles of breathing, including the tongue and the diaphragm. And the focus here in this talk, in this second segment, will be the pre-Botzinger complex, this few hundred cells in rodents that are thought to drive the respiratory network. And thirdly, the fact that there is not one nor two, but multiple sites modulating the chemoreflex, the response to hyperventilation through increases in CO2 and drops in oxygen. There are multiple chemoreceptor sites in the brain that respond to changes in CO2, that modulate the ventilatory response. The important point is that many of these cell groups are themselves modified by sleep-wake state. So at this point, I just want to emphasize that sedation-induced impairment in breathing essentially encompasses three issues: central respiratory depression, upper airway dysfunction, and reduced brain arousability. And we're going to focus mainly on the top two because of time. I think the next talk will focus very much on the clinical aspects. I just want to introduce the idea that it's the analgesic properties of the juice extracted from the opium poppy has been recognized for centuries and was written about. For example, Hippocrates was the first Westerner to write about this in medical terms, BC. And since then, these mu-opioid receptor agonists are the mainstay of pain management, because they work fantastically well. But it's been known for as long as these drugs have been available that there is an accompanying predisposition to respiratory depression, which in some cases can be serious. Such that it's considered and written about that provision of effective analgesia can be suboptimal because of the concerns in some patients about the predisposition to respiratory failure. What's a particular concern is at night when a patient may have opioids on board, deemed in a satisfactory condition, and then at a later time may fall asleep and have serious respiratory complications. And this state dependency, the ability of wakefulness to mask a potential situation of respiratory depression, is something we'll get to a bit later. And the third thing is that this is a statement from the Principles and Practice of Sleep Medicine. Caution and close respiratory monitoring is considered mandatory in sleep apnea patients post-operative, and opioids should only be administered if the airway is under control. In some cases, this is not possible, especially in situations where people they don't know they have sleep apnea, and also if at some point the patient goes home and has their opioid drugs and falls asleep at a later time, perhaps in combination with other drugs like ethanol. This individual we'll meet again later. He's post-op. This is a post from a Facebook page, and the post says, 5 hours post-op in recovery room, morphine running well. Patient's very happy. He's heavy. He has a clear phenotype of someone who maybe have obstructive sleep apnea. Now, I'm going to focus on a particular area of the brainstem that's critically involved in opioid-induced respiratory depression. And it may look as if I'm excluding other sites, but I want to acknowledge that there are multiple sites potentially where opioids could cause respiratory depression of central respiratory neurons. We'll get to all these different sites later. There's motor pools, there's inspiratory neurons of the ventral respiratory column. But I'm going to focus on the pre-Botzinger complex. The reason being is that this region is thought essential to generating respiratory rhythm in mammals. Some of the initial evidence, which has been further substantiated since the initial observations, were that if one is to selectively ablate or kill or inhibit cells in this region by chemical means, you can severely disrupt breathing, and that breathing disruption first appears in sleep. Here we have breathing movements recorded over time in a rat in a plethysmograph. And here there is an example several days later after the cells in this particular region, which is the pre-Botzinger complex, are selectively killed. And breathing becomes very ataxic and irregular. This was the first evidence that, this is very basic science work, but it was the first evidence that translated the fundamental cellular mechanisms of respiratory rhythm generation to the in vivo whole animal preparation, and identified that the integrity of the pre-Botzinger region, this cluster of cells, several hundred to 1,000 or so cells either side of the midline, is essential for generating rhythm. And it was at this point that Gaspard Montandon, who's a postdoctoral fellow in the lab, decided to develop methods to selectively manipulate this region of the brainstem reversibly in vivo in an intact animal preparation. This is very difficult. We had previous extensive experience modulating the hypoglossal motor pool, which I'll describe in later experiments, but to target the pre-Botzinger region of the brainstem, this tiny area that's modulating respiratory rhythm, is very difficult. And there's very few people worldwide who are able to do this, and Gaspard is one of them. And these are some of the papers he's published recently and that are in submission. And this one I'm going to talk a bit about today. So I'd like to acknowledge Gaspard's major contribution to this work. So he targeted the pre-Botzinger complex region of the medulla while recording diaphragm muscle activity in vivo in rats, either anesthetized or across sleep-wake states. You'll see examples of both. He did this by lowering microdialysis probes into this region of the medulla. We use microdialysis because once this probe is in place, you can just leave it there and switch between drugs of choice, either giving nothing, which is artificial cerebrospinal fluid, or switching to any drug of choice. And what this is, only the end of this cannula, where there's a semi-permeable membrane, you can diffuse drugs into this region and do it in combination and do all sorts of things. So the first thing he did was ask the question, what happens if I apply opioids to this region? And so we have, I will remind you, these are not systemic injections. These are local application of small quantities of drug to a particular region of the brain, in this example targeting the pre-Botzinger complex. And he gets a reversible, repeatable, and highly reproducible suppression of respiratory rate with local application of a small dose of a mu-opioid receptor agonist, and it's reversed with naloxone. I'll explain this correlation map later, but here we just want to identify that the hotspot region in the brainstem for opioid-induced respiratory rate depression is a circumscribed region which is characterized by local expression of neurokinin 1 receptors, so anatomically it corresponds to this region of the pre-Botzinger complex. If Gaspard upped the dose of mu-opioid receptor agonists, again applied only to this region of the brainstem where the pre-Botzinger complex is, we can see steady respiratory rate to begin with. This is diaphragm muscle activity and tongue muscle activity. You're seeing on a very compressed time scale, so you're not seeing individual breaths. Local application of mu-opioid receptor agonists at a higher dose than you saw previously, and you can shut off breathing. And this would be fatal unless rescued by mechanical ventilation and then subsequently naloxone. Let's think about this for a second. This animal has no spontaneous breathing movements at all, and that's persistent, despite the fact that over time there is accumulation of CO2, there's drops of oxygen. There's no ability for this animal to rescue breathing unless we reverse the opioid receptor effects only in one spot in the medulla. So despite all the chemoreflexes that are going to be kicking in, the presence of opioids at one site in the brainstem is sufficient to abolish breathing, and this would be fatal apnea unless otherwise rescued. Here we are applying the same interventions at a lower dose that doesn't cause serious breathing problems, and we can do this across sleep-wake states. So here we have one example of a rat in one experiment lasting a good hour and a half, I would think. And we have periods of cycling between wakefulness, non-REM sleep, and REM sleep. We have the EEG, neck muscle activity, and spontaneous changes in respiratory rate. It's high in wakefulness, less in non-REM sleep, etc. If we apply this mu opioid receptor agonist, again, only to the pre-Botzinger region of the medulla, you can see just by your eye that there is a modulation of respiratory rate, there is a depression, and it's recoverable with naloxone. But what we were interested in is the state dependency of the response. So we measured how much did rate go down, and in what state did this happen. So we have multiple epochs of EEG activity that we analyze with frequency spectral analysis. We have multiple measures of breathing, and we have multiple measurements within and between animals. And here's an example of one response in one animal. We can quantify the index of sleep depth as a ratio of high to low frequencies. In short, if the number is high, it means there's a preponderance of high-frequency activity in the electroencephalogram. If the number is low here, it means there's a preponderance of high-voltage, slow-frequency activity. And we have all these different epochs, because, of course, the electroencephalogram spontaneously fluctuates between periods of very high voltage, less high voltage, but it's still asleep, etc. And what we found was a clear relationship with some variability. But the point being that the presence of opioids at one region of the medulla, the pre-Botzinger complex, will elicit significant suppression of respiratory rate in states of sleep, this example, and we did some other experiments, in states of anesthesia, but less so, and sometimes not at all, in wakefulness. The principle here is that wakefulness itself can mask the impending respiratory depression that certain drugs can produce, in this case opioids. So there may be, in this situation, the example is the animal has opioids at its region of the brain that generates rhythm and modulates respiratory rhythmic activity. In wakefulness, you wouldn't know that, because wakefulness is providing enough of a stimulation to stimulate breathing just by itself. But as soon as that wakefulness stimulus is withdrawn, there is now the pre-Botzinger region is highly sensitive to opioids, and in states of deep non-REM sleep and anesthesia, respiratory rhythm crashes. So what is this hotspot region, and how do we find out where the region was that's highly responsive? So, well, the pre-Botzinger region is very hard to target. So you sometimes get further away from it than you aim, but that's a good thing, because you can use that information to productive value. So in every animal, we knew when we applied, in this case, fentanyl, we knew the responsive rate, we knew how long it took for the drug to travel down the tubes, and we could quantify the latency to, in this case, a 10% decrease in respiratory rate. And we could do this in every animal. And what we could do then is quantify how far the probe was from a particular spot in the medulla. In this case, I've just drawn it for simplicity to the pre-Botzinger complex as defined by neurokinin 1 receptor activity. And we can get a correlation between the latency of response to a decrease in rate and the distance. But a priori, we decided to do this without deciding that this pre-Botzinger region was the site of opioid-induced respiratory depression. So we divided the medulla into a 50-micron grid and did multiple correlations to every possible site. And what you do then, you can get R values and R squared values for a grid of the medulla without presupposing where the most responsive site is. And so you get multiple R values, which we can then plot as a heat map with respect to its magnitude, 1 being very high and red, and blue being very low and 0. And this is our hotspot map. And if we filter the correlations to only look at those that are above 0.8, the hotspot region for this opioid-induced respiratory depression is slap bang in the pre-Botzinger complex region of the medulla, right there. We went further to address further mechanisms of action, so we collaborated with John Greer at the University of Alberta, who's developed slice experiments. You can do much more detailed and sophisticated neurophysiology on these cells, and he pre-labeled pre-Botzinger complex neurons with a red dye, and this rhodamine is only present in those cells which are neurokinin 1 positive. Now, I've mentioned neurokinin 1 before, but now I'll mention why this is important. So the neurokinin 1 receptor is the receptor for substance P, and it's considered an anatomical marker for pre-Botzinger complex neurons that are rhythmogenic. So these are the ones that are thought to provide the rhythm of breathing. There are non-neurokinin 1 receptors too, but they're not thought to be the ones that generate the pacemakers for breathing. So in collaboration with John Greer at the University of Alberta, we measured hypoglossal nerve activity. This just gives you an indication of rate from the whole nerve. And this is a single cell recording from a neurokinin 1 receptor expressing neuron. These are individual respiratory drive potentials, and in the presence of the mu-opioid receptor agonist DAMGO on this slice of medulla that contains the network for rhythm generation, you can see the cell is hyperpolarized and loses all rhythmicity. This seems to be why opioids are depressing rhythm. And it's important because if you do the same thing on cells that are non-neurokinin 1 receptor expressing, you see no response. So the phenotype of the cell that's inhibited by opioids is neurokinin 1 receptor expressing neurons in the pre-Botzinger complex region. Just because this region is responsive to opioids doesn't mean to say it's critical. And I think this was the powerful experiment that proves that it is, of all the potential sites that are operating in medulla, that the pre-Botzinger complex is perhaps the most sensitive. So here we have a situation where we have a rat with the microdialysis cannula into the pre-Botzinger region of the medulla, and it's being perfused with artificial cerebrospinal fluid, i.e. it's a control solution. We can then systemically inject opioids at an amount that will then fill up the animal, as it were, okay? And you get a robust suppression of respiratory rate. But of this, the opioids could be acting anywhere. So how important is the pre-Botzinger complex? Well, in this situation now, we have naloxone only applied to the pre-Botzinger region of the medulla. And we can then, again, give a systemic injection, which again fills up the animal, and it could be acting anywhere. The important point is, though, of all the potential sites that opioids could be influencing, the peripheral chemoreceptors, ventral respiratory group neurons, retrotrapezoid, raphe, if you apply the antagonist only to the pre-Botzinger region bilaterally, you prevent all respiratory rate-induced suppression. So of all the potential sites of action of opioids, it seems that at least in this mammalian preparation, that the pre-Botzinger region is the most sensitive and important. Doesn't explain how opioids are working to depress rate, and this is what we'll move to now. The mu-opioid receptor can mediate, hypothetically, the depression of rate through one of several mechanisms, including modulation of N-type calcium channels, modulation of adenylate cyclase. One of the, through various literature searches, we decided that we would focus on this potential modulation of the G protein inwardly rectifying potassium channels. The idea being that the mu-opioid receptor, once stimulated by the mu-opioid receptor agonist, will open this channel, causing potassium efflux and cellular inhibition. There are several confirmations of the GERK channels, GERK 1 and 2, 1 and 3, 2 and 3, and 2 and 2. Through a collaboration with Kevin Wickman, we got a breeding colony of GERK2 knockout mice, and so we could remove 3 of these receptor complexes simply by knocking out the GERK2 receptor, GERK2 subunit. And I provide evidence to you, it's that particular complex, the GERK mu-opioid receptor, the complex, that is the cellular mechanism by which opioids depress rhythm. So first, some immunohistochemistry identified that GERK channels are co-expressed with these neurokinin-1 receptors at the Prebotzinger region. So we have a low-resolution, some high-resolution images, but a broad space, including the Prebotzinger complex, and very high, as you can see, individual cells here. The point being that we can label neurokinin-1 receptors at the Prebotzinger region. These are the cells that generate rhythm. We identify, and this is the first time it's been demonstrated, that GERK2 receptors are on those in that region too, and they're on the same cell. And you can see individual cells co-labeled. The next thing Gaspar did was he then applied an activator of GERK channels, causing them to open, and there was a depression of rate. And then in the presence of that same agent that opens the channel, in the presence of a blocker, and this is a barium at a particular concentration, blocks inhibitory rectifying potassium channels, you can restore rate. And the hotspot region for this, again, encompassed the Prebotzinger complex. Most interesting, he applied mu-opioid receptor agonists directly to the Prebotzinger region, and you can see this profound suppression of respiratory rate in normal mice, i.e. they have intact GERK subunits, these are called wild type. The baseline starting rates are shown here, so there's profound suppression of respiratory rate in the normal mice, and in the GERK2 knockout mice, no response. So it seems that the respiratory rate depression with opioids in this critical region is GERK2 subunit dependent. It's not that these cells are unresponsive to other things. For example, we gave a GABA-A receptor agonist, and you can see there is clearly the ability to suppress rate in these knockout mice, just not through opioids. And it's not that the neurokinin-1 receptors themselves are impaired in any way, because if we apply a neurokinin-1 receptor agonist, we can stimulate breathing in a normal fashion in both the wild type and the knockout mice. So the reason that they have this abnormal depression of rate is not because there's a dysfunction of the receptor itself. There's no otherwise abnormality in the cells and the machinery, the generating rhythm, is that they just don't have the GERK2 subunit, which is therefore essential for how opioids are depressing breathing. And if you give systemic opioids in these wild-type mice, again, you can produce apnea that's persistent and powerful, but in the GERK2 knockout mice, much less so. Group data is shown here. Respiratory rate the opioid-induced depression of rate is markedly attenuated in these GERK2 knockout mice. How can you use this knowledge to modulate breathing? Well, we can re-stimulate breathing in the presence of opioids by modulating potassium channels, either non-selectively or what I'll show you in a second, selectively. Here you can see a trace showing normal breathing, an example, normal diaphragm activity. This is an anesthetized animal. We give a mu-opioid receptor agonist, depression of rate, which we can rescue by giving a blocker of potassium channels, in this case barium, which blocks inwardly rectifying potassium channels. But there are very selective GERK channel blockers, and this is tertiapin-Q, which is it's in bee venom. And again, you can rescue opioid-induced respiratory depression by specifically targeting GERK channels by blocking them. So I'll conclude this section by indicating that, summarizing all I've just said, that opioids at the Prebotzinger complex region of the brain depress rhythm, and the Prebotzinger region, again, is of interest because this is thought to be the origin of the rhythmicity of breathing that imparts rhythmicity to other areas of the medulla that generates the whole network activity. But some people consider that the pre-Botzinger region is the initiator of these events. The opioids in this one region can be lethal unless rescued. And the depression occurs most in sleep, and I didn't show you, but also anesthesia compared to wakefulness. So wakefulness can mask the effects of opioids in this region. The hotspot for the effects is the pre-Botzinger complex. The neurokinin 1 receptor expressing neurons are responsible. In this region, GERK channels are co-expressed with the neurokinin 1 receptors, and they mediate largely the rate depression. And the challenge is, what can we do with this knowledge? And one of the things is to perhaps devise strategies to reactivate breathing when it's otherwise depressed or failing. There are other strategies to do so. This was an original paper in Science. It's showing that serotonin type 4 receptors are largely only expressed, 4A receptors, largely only expressed in the pre-Botzinger region, and stimulation of these receptors can essentially, it doesn't address the mechanism by which opioids depress breathing, but it basically, if this side of the pathway is depressed, if you stimulate this side of the pathways, you just trick the cells into reinstituting the downstream events, and you can reverse opioid-induced respiratory depression without affecting analgesia. This is not yet translated into clinical practice. There's a very recent study looking at another component of this same pathway. These modulate cyclic AMP. And there was some talk yesterday about ampakines, which are ways to increase the glutamate transmission at the cells that drive breathing and keep it going in the presence of opioid drugs. And these are actually, the data is, has been translated from rodent animals, multiple models, now to initial trials, phase 1 in human subjects. So I'd just like to move on now to the upper airway. We've really focused so far on the rhythm generation, but now we're going to talk about upper airway dysfunction in the presence of opioids, but also in sleep. I introduced this idea before. You can utilize the different probe placements that you can find to measure the latency of response to a decrease in rate and construct correlation maps which reveal the hotspot region for opioid-induced respiratory depression, which, as I mentioned before, is very much centered on the pre-Botzinger complex. But you'll notice down here that we also saw depression of upper airway muscle activity. And it's interesting, we correlated the latency and the magnitude of response for the genioglossus motor suppression, and it revealed a different hotspot region. Again, as I mentioned before, there's no a priori assumption of where the hotspot region might be, because we did these correlation maps for the whole region of the medulla, and we came up with a different hotspot region. This hotspot region is anatomically different, and it corresponds to, I'll mention this again a bit later, it corresponds to the cells that are intermediate between the pre-Botzinger complex, this is the region of the reticular formation, and the idea is that from anatomical work is that you've got the rhythm of breathing in the pre-Botzinger complex here, which then drives cells in this region, which then impart rhythmicity and activation to the upper airway musculature, in this case the hypoglossal motor pool. And it turns out that this same region, which is the premotor site for the hypoglossal motor pool, is the hotspot region for opioid-induced hypoglossal motor suppression. This is just schematized in the 3D version here. So with respect to network activity, we've got the idea that the pre-Botzinger complex region is opioid sensitive, and through its connections can depress diaphragm activity. But this pre-Botzinger region drives cells in the pre-hypoglossal region, which I've just shown you is also opioid sensitive. But it also turns out that the third component of this circuit, pre-Botzinger through the pre-hypoglossal, the third component is the hypoglossal motor pool itself. And so we've done a lot of work over the years in the control of the hypoglossal motor pool, and this region is itself very sensitive to opioids also. And if you apply opioids in this region, you would expect, if it's a responsive site, that the tongue muscle activity would decrease, because this is where we're applying the drug, but now we're well away from other areas, and the response is very selective to the tongue musculature, as you would expect, showing that even though there's distribution of drug, It's really only affecting, in this experimental situation, the upper airway. So, to conclude how opioids are acting, the key components of the respiratory network mediating respiratory depression haven't focused on areas involved in chemoreception and arousal centers, but the prebotzinger complex, the parahypoglossal nucleus, which is the source of hypoglossal premotor neurons, and the hypoglossal motor pool are all opioid sensitive. There are other sites. The key thing I want to mention, though, is I think that experiment with naloxone in the prebotzinger complex suggests that the prebotzinger complex is hypersensitive. It's not to the exclusion of other sites that may be also responsive. So I'm just going to finish up with some, in the next 10 minutes or so, with some recent work on the upper airway performed by Kevin Grace, who is a PhD candidate, PhD student in the lab, whose thesis is on mechanisms of REM sleep generation and respiratory depression. Kevin, there's some very interesting work now looking at how REM sleep is generated, and well, perhaps this is the way science works. There's been camps that have pitted themselves against each other with respect to whether there is a cholinergic, aminergic mechanism of REM sleep generation or a glutamic GABA mechanism of REM sleep generation. Kevin has done some work, actually recently that we're submitting this month, showing that these actually work together to fine-tune the transition to sleep-wake state. It's not one or the other, they work together, and together they optimize transitioning and maintenance of sleep-wake states. What I want to focus on, though, is the control of the spinal musculature in REM sleep. And the motor inhibition, whichever circuit you look at, is caused by a release of glycine and GABA, which causes a powerful inhibition of the spinal motor pools. And I think this evidence is very solid. What is not solid is what's happening to the upper airway in REM sleep. And I'm going to propose to you that this mechanism is not operating, the glycine and GABA, to any appreciable extent, and in fact that there is a cholinergic inhibition of the upper airway musculature in REM sleep that's very powerful. I'm going to run through this somewhat briefly, but I want to give you a flavor of where we've been at. So the idea is that motor pools, well, now we're talking about the final common output pathway to the respiratory musculature. And essentially, motor pools receive excitatory and inhibitory inputs. And the motor neuron sums these inputs, sees what they're at, and produces a response accordingly. The idea has been, as I just mentioned, that there is release of glycine and GABA to motor pools in REM sleep, and this is responsible for the inhibition. As I mentioned, I think this is exactly right for the spinal musculature. I do not think it's appreciably significant for the upper airway. Some evidence that indicated that said that motor neurons that drive the tongue musculature do receive inhibitory potentials in at least a pharmacological model of REM sleep. This is membrane potential of the hypoglossal motor pool, and this is in a pharmacologically induced REM sleep, which I won't get into because of time. You can record these inhibitory potentials, which you can block with a glycine receptor antagonist.